Assalamu alaikum. Welcome to the great biochemistry lectures in the hematopoietic-lymphatic system. So in this lecture what we will do is we'll talk about metabolism in erythrocytes, we'll talk about the major metabolic pathways and their significance to the cells and to the system as a whole. So there are two metabolic pathways that are important in red blood cells. The first is glycolysis and the other is the pentose phosphate pathway. Each one of them has a purpose. The purpose of glycolysis is to produce 2,3-bisphosphoglycerate and to produce NADH as well as ATP, by the way. Now the second pathway, the pentose phosphate pathway, the main purpose is to produce NADPH. Okay, now, so let's start with the 2,3-bisphosphoglycerate. You already know that 2,3-bisphosphoglycerate binds to the center of the hemoglobin molecule, stabilizing the T state of the molecule, reducing affinity towards oxygen, right? So, where does 2,3-bisphosphoglycerate come from? It comes from the glycolytic pathway. So what happens is glucose is eventually broken up into two molecules, glycerol 3 phosphate and dihydroxyacetone phosphate. Glycerol 3 phosphate continues with the pathways, producing 1,3-bisphosphoglycerate. Eventually, this is converted to 3-phosphoglycerate. But there is a byproduct, which is the 2,3-bisphosphoglycerate. It is an isomer. Now remember something, by the way. Notice that if you convert 1,3-bisphosphoglycerate to 2,3-bisphosphoglycerate, this molecule, the 2,3-bisphosphoglycerate, can be converted to 3-phosphoglycerate. Okay? But there is a loss of ATP in these pathways. So this is a disadvantage in this pathway, but the advantage is that you do have some 2,3-bisphosphoglycerate in red blood cells to allow for the release of oxygen from hemoglobin in tissues. Okay, so what 2,3-bisphosphoglycerate does is that it binds to the center of the hemoglobin molecule, interacting with several amino acids around it. Since it is highly negatively charged, so it interacts with possibly charged molecules like histidine, for example, and lysine. Okay? Now, what happens is that by forming these electrostatic interactions, the binding of oxygen to hemoglobin is reduced. So you have stabilization of the T state. So that shows the normal concentration of, that shows the oxygen saturation curve in the presence of normal physiological concentration of 2,3-bisphosphoglycerate. Increasing the concentration of 2,3-bisphosphoglycerate from 5 millimolar per liter to 8, that shifts the curve to the right, meaning that affinity is reduced. If there is no 2,3-bisphosphoglycerate in cells, that would increase the affinity very much, so it looks almost like a the oxygen saturation curve of myoglobin. Okay. Now, what happens, what's really important here is that 2,3-bisphosphoglycerate does not interact very strongly to the fetal hemoglobin. The reason is that in fetal hemoglobin, instead of histidine 143, which forms electrostatic interactions with the 2,3-bisphosphoglycerate, there is no histidine 143. Rather, in fetal hemoglobin, you have a serine. So that weakens the interaction between 2,3-bisphosphoglycerate with hemoglobin, increasing the affinity. So this is a reason why fetal hemoglobin has higher affinity towards oxygen than adult hemoglobin. So this is another view, and a third one. So you can see the interaction between the 2,3-bisphosphoglycerate with histidine 143. So this is gone. replaced by serine. Same thing here. Okay, so there are different views of the same idea. So let's talk about the glycolytic pathway. Now, why is that important for red blood cells? It's important because both NADH and ATP are produced. So NADH is important for reoxidizing methemoglobin. So if you remember, we talked about that in the previous lecture, and we said that cells use a cytochrome containing enzyme, a reductase enzyme, to convert methemoglobin to hemoglobin. Now, ATP is important for red blood cells for several reasons. One, it is used for first the glycolytic pathway, of course, the initial reactions. It's important for modifying sugars and proteins. It's important for maintaining membrane asymmetry. It's important for the function of ion pumps, and it is important for modifying and regulating cytoskeletal proteins. These proteins that are important for changing, altering the morphology of red blood cells. So what they do is that they maintain the discocytic shape. This is the discocyte shape. That's how it looks like. Okay, and it's important for the function of these proteins so that red blood cells can squeeze in the narrow capillaries to reach tissues. So this is the glycolytic pathway. Glucose is ultimately converted to pyruvate. The very last reaction is catalyzed by pyruvate kinase, and what it does is that it phosphorylates ADP, producing ATP, converting phosphoenol pyruvate to pyruvate. So let's talk about pyruvate kinase now. Now, pyruvate kinase, there are two isoenzyme genes. So there are two genes that produce pyruvate kinase, and each one of them can produce two isoforms. The genes are known as PKLR gene, and the other one is known as PKM gene. Now, PKLR, L stands for liver, and R stands for erythrocytes. So this gene can produce two different isoforms of the enzyme, PKL and PKR. The difference between them is that there are different transcription start sites. Okay. Now, PKM, where M stands for muscle, is produced. There are two isoforms, PKM1 and PKM2. PKM1 is mainly produced in muscle and brain. PKM2 is produced in fetal tissues, as well as all other different types of tissues. Now, the thing about PKM2 is that it has much greater activity than the adult isozyme. Okay, so that's really important. So that gives the fetal tissues and fetal red blood cells an advantage right here. Now, since pyruvate kinase is very active in erythrocytes, that would lead to reduced amounts of glycolytic intermediates, including 1,3-bisphosphoglycerate and 2,3-bisphosphoglycerate. So the pathway is moving very fast. So the intermediates are at low concentration in fetal tissues, okay, including erythrocytes. So this is really an advantage right here. Since fetal tissues have lower 2,3-bisphosphoglycerate, that is red blood cells or erythrocytes, that would mean that fetal tissues would have higher affinity towards oxygen because there would be high proportion of fetal hemoglobin being in the R state. Now, pyruvate kinase is also differently regulated in different tissues. So the PKL and PKR isoforms are allosterically regulated. So they are activated by fructose 1,6-bisphosphate, and they are inhibited by high levels of acetyl coenzyme A, ATP, long-chain fatty acids, and alanine. So these are the sources of energy. If they are in high levels, the enzyme is inactivated. Now, the enzyme is also regulated by phosphorylation by protein kinase A. And if there is high level of glucagon in the system, that would ultimately activate protein kinase A, which would phosphorylate pyruvate kinase. Now, the liver enzyme, by the way, is also regulated at the level of synthesis. So if there is high level of carbohydrates in liver or in the system, that would induce the synthesis of protein kinase, pyruvate kinase, I'm sorry. There are conditions related to pyruvate kinase deficiency. These conditions are genetic, so they are hereditary. And in these conditions, the enzyme is not really that active. Mainly these are caused by single point mutations, and what they would do is that they would reduce the ability of erythrocytes or red blood cells to produce ATP, resulting in hemolytic anemia. Now, just like any other inborn error of metabolism or enzymatic enzyme-related diseases, the severity of the disease depends on how much the enzyme is deficient. So if an enzyme, for example, for many enzymes, if it is, let's say, 50% deficient, the individual should be fine. But if it is less than that, if it is like in case of pyruvate kinase, if it is 5 to 35% of normal activity, then symptoms would appear. Now, the thing about liver is that it is not affected since expression is stimulated, but erythrocytes would be affected. Now, enzyme activity can be reduced as a result of either reduced activity itself, that is, it cannot bind to the substrate, or it cannot bind to the inducer, the activator fructose 1,6-bisphosphate, or it could be due to decreased stability. So the amount of enzyme in cells is reduced. So let's talk about the second pathway now, the pentose phosphate pathway. Well, the pentose phosphate pathway is divided into two phases. You have the oxidative phase and you have the non-oxidative phase. The main purpose of the oxidative phase is to convert glucose 6-phosphate to ribulose 5-phosphate. Eventually, this would be converted to ribose 5-phosphate. But the main purpose of these reactions is to produce NADPH. Now, the non-oxidative phase of this pathway is to convert ribulose 5-phosphate to different types of sugars, including glucose 6-phosphate. So, the most important reaction in the pentose phosphate pathway is the very first reaction, which is catalyzed by the enzyme glucose 6-phosphate dehydrogenase. This is a rate-limiting reaction, and it is irreversible, and it is highly regulated. Now, this enzyme has higher affinity, is very specific to NADP relative to NAD. So, and it is regulated by NADP, so high levels of NADP would increase the activity of the enzyme. Now, why is it that NADPH is important? Because it is responsible for regenerating glutathione. Remember, glutathione is a small peptide molecule. It is made of 3 amino acids: glycine, cysteine, and glutamate. Now, in the presence of hydrogen peroxide, what happens is that two glutathione molecules reduce hydrogen peroxide into oxygen and water, producing the oxidized glutathione molecule, where these two molecules are connected to each other by a disulfide bond. Now, in order for red blood cells or any other cell to fight more of these oxidizing agents, the oxidized glutathione must be regenerated, and it is regenerated by the enzyme glutathione reductase. The source of electrons is NADPH. So electrons are taken from NADPH to reduce the oxidized glutathione, producing these two molecules. And the pentose phosphate pathway is the main source of NADPH in red blood cells. In fact, the pentose phosphate pathway consumes about 10% of glucose in erythrocytes. What if you have low glutathione levels in cells like erythrocytes? Well, what happens is that there would be high levels of hydrogen peroxide in cells. That would cause weakening of the cell membrane and hemolysis of red blood cells. So the membrane would be compromised because hydrogen peroxide would start to oxidize fatty acids in the plasma membrane. Now, what also happens is that there would be higher level of methemoglobin as well, because the rate of oxidizing iron would be high as well. Now, what will happen in this case is that the hemoglobin molecule, predominantly, as well as other molecules, would start to become insoluble, and they aggregate, forming what is known as Heinz bodies. And you can see these Heinz bodies in these cells right here. So there is a condition or group of conditions related to deficiency of glucose 6-phosphate dehydrogenase. Now, these deficiencies collectively, what they do is that they cause hemolytic anemia, because again, plasma membrane is compromised by high levels of hydrogen peroxide as a result of low levels of glutathione. Now, this hemolytic anemia can be induced, or it can become more severe if certain drugs are administered, or as a result of infections or jaundice. Now, what's important about G6PD is that deficiency is that it is very prevalent in the old world. So you can see how the distribution of this disease globally, and there are different types or classes of the disease, and we'll talk about them in the next slide. Now, the gene for G6PD is on the X chromosome, so inheritance pattern follows sex-linked diseases. Again, this is the distribution of the different types of glutathione 6 phosphate dehydrogenase deficiency, and note the distribution of a certain type in the old world in the Middle East. So, deficiency of G6PD is related to single point mutations rather than deletions or frame shift mutations, and that tells you something about the importance of this enzyme in not only for erythrocytes and red blood cells, but also for the whole individual. So these mutations can affect the kinetic properties of the enzyme, the stability of the enzyme, or the ability of the enzyme to bind NADP or glucose 6-phosphate. Now, there are different variants of the enzyme G6PD. Of course, you have the normal variant with 100% activity. And then you have different variants. These variants can be classified into 4 classes. So class 1 is basically variants that have very little activity of G6PD. That would result in very severe condition of G6PD deficiency, resulting in chronic hemolytic anemia. Now there are many variants that are classified as class 4, and these variants have high activity of G6PD with no symptoms whatsoever. Now let's focus on these two, class 2 and class 3. Now class 3 variants are mainly African type, so they are more common among people of African descent. Now what happens here is that you have again single point mutations. Primarily you have a mutation changing asparagine to aspartate that would decrease enzyme stability, but the enzyme is still active. Now the activity of the enzyme is about 5 to 15% of normal activity. The disease is moderate, okay, but it becomes severe under certain conditions, including what we'll talk about right now in a second or so. So class 2 are known as G6PD Mediterranean because they are common among Mediterranean individuals. Now here you have normal stability of the enzyme but very little activity, and it's a severe condition. So let's look at a little more closer on class 2 and class 3 conditions of G6PD deficiency. Now in normal individuals, this is the age of erythrocytes, and as red blood cells age, the activity of G6PD is reduced. Now for the African descent G6PD deficiency, or class 3 or A minus, the enzyme activity is high. Yes, it gets reduced as erythrocytes age, but it's still high. That's why the condition, or the severity of this condition is moderate. But if you look at the Mediterranean type of G6PD deficiency, although enzyme stability is high, there's hardly any activity, and it gets reduced early on as erythrocytes age. So there are inducers for symptoms of G6PD deficiency. People with this deficiency taking oxidizing drugs like antibiotics, antimalarial, antipyretics other than acetaminophen can exacerbate symptoms related to G6PD deficiency. Ingesting fava beans, a condition known as favism, can also induce G6PD symptoms. Now fava beans are فول بالعربي. So why is it that? Because fava beans contain substances that can destroy glutathione, reducing the amount of glutathione in cells, so that would again reduce the half-life of red blood cells. Favism is very common among class 2 variants, that is in the Mediterranean type of G6PD deficiency. It rarely occurs in patients with the African class of G6PD. Okay. Now infections can also induce G6PD deficiency symptoms as a result of production of oxidizing agents by immune cells. Finally, G6PD deficiency is associated with resistance to malaria, and the reason is the age of red blood cells is reduced, preventing the completion of the development of this parasite. So you can see the overlap between the prevalence of G6PD deficiency in the Old World, in Africa, the Mediterranean region, including the Middle East, our countries, including Jordan, and prevalence of malarial infection.